Hello students, welcome to the new lecture, and in this lecture we are going to discuss about the cell wall. So in the previous lecture we have discussed some points, and now we are continuing with that. So in this chapter, this is the second lecture, okay, the cell wall. So what is meant by cell wall? So cell wall, it is nothing but the outermost layer, okay. So let us consider this is a cell, okay. The cell contains cell membrane. This is cell membrane. Around the cell membrane there is a cell wall, okay. So this cell wall is the outermost layer in case of plant cells, but in case of animal cells, there is no cell wall, only the cell membrane is there. Okay, that we will discuss after a moment. And the cell wall is the outermost layer in plant cells, but in animal cells, the cell membrane is the outermost layer. Outermost layer means outside layer. Okay, outermost layer means outside layer, that is known as outermost layer. So the cell wall in plants is made up of cellulose. Okay, what is cellulose? It is a material. Okay, so cellulose, it is a material. This cellulose is utilized for the production of the cell wall. Okay, in the plants. Okay, so in animal, in bacterial cells, some other component is used. But remember, in case of plant cells, the cellulose, it is the material used for the production of outermost layer, that is nothing but the cell wall. Okay, so here the cell wall is tough. For example, let us consider the animal tissue. So this is the animal tissue. What is meant by tissue? The tissue is nothing but the group of cells. Okay, the group of cells with similar size, shape, and function. Okay, these cells have the same size, same shape, and same function. This is known as tissue. Let us consider this is the animal tissue. Okay, so and now we will take the plant tissue. Okay, the plant tissue. So the same. Okay, which one is more softer? If you touch the plant tissue, does it look softer or harder? Okay, the plant tissue look harder, okay, and tough. And the animal tissue, if you touch the animal tissue, it is soft. Okay, the animal tissue will be soft. So the reason is, the main reason is, one of the reason is the cell wall. Due to the presence of the cell wall, the plant tissue is a little bit tough, and it is going to provide toughness, and also at the same time it is flexible. Okay, if you bend the plant, in most of the plant, if you bend the plant will come to the normal position after you release the pressure. Okay, next, this is cell wall. Not only that, not only tough and flexible, but also it is going to produce, it is going to provide the porous means bit. Let us consider this is the cell membrane. So this is the cell wall of a plant cell. It has small holes. These small holes are known as these holes are known as porous materials. Okay, it means the cell wall is made up of small holes. This is known as porous. So due to the presence of small holes in the cell wall, we are calling the cell wall as porous. Okay, this is porous, and the cell wall is going to provide the protection. So how it is going to provide protection? The plants do not move from one place to another place, so they have to withstand higher temperature, lower temperature, rain, wind, etc., all these things. So to get protected from all these factors, to get protected from all these factors, the cell wall is most important. The cell wall is most important. Hence we can say that the cell wall is going to provide protection for the plant cells. Okay, so it is also going to exchange the information during the growth and development. So with through the cell walls, let us consider this is a one cell wall of a one plant, one cell. So this is cell wall of another cell of a plant. This is cell membrane, and so on. It means information is getting exchanged from one cell to another cell through the cell wall. So during the growth and development, it is one of the important function. Okay, so this is about a cell wall. And now we will see what is the difference between cell wall and cell membrane. So look at this diagram. This diagram, we can see that the cell membrane is there. So this tough layer is known as cell membrane. Okay, this is a tough layer is known as cell wall. Okay, so beneath the cell wall, a small layer like structure, so a light blur structure is there. Now this one will be the cell membrane. Okay, so cell membrane and also cell wall, both are present one after the other. First cell wall will be there. So this is a cell wall, complete one is a cell wall. After the cell wall, a thin layer, this thin layer is known as plasma membrane or cell membrane. Plasma membrane or cell membrane, both are same. So this is a plant cell. So this is only the reason we are able to see the cell wall. Okay. So cell wall along with the cell membrane that can be seen in the cell plant cell. Okay. So now we have discussed about the importance of the cell wall. So then what is the functions of it? So the cell wall is going to provide the wall pressure. Okay, the wall pressure is one of the main important factor. So let us consider where do this wall pressure is more important. So let us consider this is a cell, the plant cell. Okay, so the plant cell containing the cell wall and also cell membrane. Okay, so what will happen, the water during the rainy season, the more water is there, okay, the water will enter into the cell. As a result, what will happen, the water will get accumulated inside the cell. Okay, the cell cytoplasm has more amount of water. It is going to exert pressure, it is going to exert pressure on outside. So to withstand this pressure, to withstand this pressure, what should be there, there should be some supporting material. That supporting material is known as a cell wall. It means this cell wall is going to provide it is going to provide wall pressure. It means at the same time this cell wall is going to provide against pressure to the onto the cell membrane. So such kind of exertion of the pressure by the cell wall is known as wall pressure. So now the case is, now let us consider there is no cell wall. There is no cell wall, only cell membrane is there. What will happen? More water is there. Okay. So more pressure, the more pressure will be built inside the cell membrane. As a result, it cannot withstand, it will burst. Okay, it will burst. It means the cell wall will burst. As a result, the plant will die. Okay, so this is one of the drawbacks. So due to this, to prevent such kind of disaster, so there is cell membrane, and above the cell membrane there is cell wall. Okay, so this is one of the function. And cell wall provides a greater chance for the survival in nature. It is going to provide the greater chance. As a result, the plants are going to survive better in the nature. Okay, survival means what? Survival means living. Okay, so now we are going to discuss about the nucleus. Now just now we have discussed about the cell wall. Okay, now we are going to discuss about the nucleus. So what is meant by nucleus? Nucleus is nothing but, let us consider this is a cell. So inside this is cell membrane. Okay, cell membrane. Inside the cell membrane there is cytoplasm. In the cytoplasm we have discussed already. So the cell organelles are there. The central largest cell organelle is the nucleus. It is the nucleus. Okay. So nucleus, here will be the mitochondria, it is the Golgi apparatus, okay, endoplasmic reticulum and so on. So these all cell organelles are there. So among all these cell organelles, now we are going to focus only on the nucleus. We are going to focus only on the nucleus. So in this experiment, we have to understand, we have to identify the nucleus in the cell. So our aim is what? Our aim is to observe the nucleus in cheek cells. So what is meant by cheek cell? Cheek cell is nothing but inside the mouth, inside the cheek wall, inside the cheek. So there is some cells, a layer of the cells. So these layer of cells inside the cheek, that is known as cheek cells. It means we have to observe the nucleus inside the cheek cells which is present in our mouth. So to observe that, we need some materials. Those materials are a toothpick or ice cream spoon or a spatula. Spatula is nothing but a spoon like structure, spoon like material. So next, glass slide, cover slip, watch glass, needle, blotting paper, 1% methylene blue, normal saline, and also glycerin, microscope, etc. So these all are the materials that are needed and necessary. Okay, so procedure, how we have to do the experiment, that is known as procedure. Procedure means method. So first we have to wash our mouth. Okay, first wash our mouth, so that if any food particles are there, they will be eliminated. So that is the reason you have to wash your mouth. Then take the spoon or spatula or ice cream spoon and scrape means slightly apply little bit pressure and drag outside. So as a result, the lining, let us consider this is a cheek. Okay, this is the cheek. So with the help of spoon, slightly drag it outside. So as a result, at the end of the spoon, what you will see, small tissue will be there at the end. So take this material, So this material is nothing but your cheek cells that is attached to the spoon. So now take that, and also you have to you have to take this material and place it on the slide. Okay, so place it on the on the watch glass. Let us consider this is the watch glass. What is meant by watch glass? Watch glass is nothing but a small glass plate. In the laboratory you will see a small glass plate. This is known as watch glass. So tissue put the tissue of a cheek cells inside the watch glass, and pour methylene blue. Okay, so pour small amount of methylene blue, and as a result what will happen, these cells will take some amount of dye. Okay, so small quantity of saline, also you have to put some small amount of saline is nothing but salt water. Okay, so then place the material. Now take out this material. Okay, and put it on a slide. So this is a slide. So on the slide put the cells on the slide, and after that what you have to do, we have to add a drop of methylene blue. Okay, drop of methylene blue. What is methylene blue? Methylene blue is nothing but a dye, a color. Okay, add a drop of methylene blue, wait for a minute, or wait for 5 minutes or 6 minutes, then cover it with, put a drop of glycerin. Okay, put a drop of glycerin. Okay, after putting the glycerin, what you have to do, put a cover slip. Okay, cover slip is nothing but a small glass plate, very thin glass plate. Put on it, and if there is any excess water, or if there is any excess dye, what you have to do, we have to clean it with the help of blotting paper or tissue paper. You have to clean it. So now that slide will be ready. The slide is ready for the observation. So take the slide and put it under the microscope and clearly observe it. If you observe the slide under the microscope, you can see structure of the cheek cells. Okay, how the structure of the cheek cells will be there, how they are going to look, you can clearly see. Okay, so here you have to wipe out the stain is nothing but a color. So extra stain which is there, you have to wipe out. So this will be the cheek cell. Okay, here you can see this will be the cheek cell. So under the microscope you can see the structure of the cheek cell, and outside layer, can you see now, outside layer, this layer is known as a plasma membrane or cell membrane. So inside the cell membrane there is a cytoplasm. So the cytoplasm is nothing but semi-solid fluid. Okay, that is present inside the cell. In the cytoplasm, all cell organelles are present. So this is cell wall. This is cell membrane. So this is cytoplasm. So the cytoplasm, inside cytoplasm, the cell organelles are there: nucleus, mitochondria, everything. So here the small dot, blue color dots, whatever the blue color dots you can see here, this is known as nucleus. Okay, this is known as nucleus. So this nucleus is present in the almost center in the cell. Okay, so by this way you can observe. So mainly our aim is to what? Our aim is to observe the nucleus. Yes, we can see the nucleus over here. The nucleus is present over here, we can clearly see. Okay, so this is about observation of nucleus in the cheek cells. So here you can see the nucleus is going to look like this. Okay, so nucleus is going to look like this. If you see here, if you take out the nucleus and if you draw clearly, the nucleus will look in this way. So the nucleus is going to have a membrane. Nucleus is also going to have the membrane that is known as nuclear membrane. So inside the membrane, there is a nucleolus. Okay, this is genetic information. This is DNA. Okay, genetic information looking like a thread. And to the membrane, there is small holes. The small holes are present on the membrane of the nucleus. These holes are known as nuclear pores. Nuclear pores are nothing but small holes, microscopic holes which is present on the nuclear membrane is known as nuclear pores. Okay, so this is about nucleus. And inside the material, the fluid like material inside the nucleus is known as nucleoplasm. Cytoplasm means what? So this is the cell. Okay, this is the nucleus. From here to here, from the nucleus to the cell membrane, there is cytoplasm. Okay, cytoplasm. So inside the nucleus, there is nucleoplasm. Okay, so nucleoplasm means the fluid like substance which is present inside the nucleus, that is known as nucleoplasm. Cytoplasm means the fluid-like substance which is present between nucleus and the cell membrane is known as cytoplasm. Okay, so this is only the difference between the nucleus and the nucleoplasm, cytoplasm, and so on. So precautions. Precaution means what care you have to take, what care you should be taken. So those are nothing but precautions while doing the experiment. So do not scrape the cheek cells too hard. If you scrape the cheek cells too hard, you are going to damage your buccal mucosa. Buccal mucosa means the layer inside the mouth is known as buccal mucosa. Okay, the layer is there, that layer of tissue will be broken, will be damaged. As a result, you will feel pain. Sometimes you may see the blood coming out. Okay, so as a result, do not scrape the cheek too hard. Okay, scrape the cheek very lightly, inside the cheek very lightly. Okay, scraped material should be spread uniformly onto the slide. So spreading uniformly on the slide is nothing but this. Let us consider this is a slide. Onto the slide, spread the cells uniformly. Okay, uniformly. You should not spread like this. You should not spread cells like this. Okay, what will happen if you spread the, if you make the cells as a small group? In that case, we are unable to observe under the microscope clearly. Okay, so excess stain should be drained off after pouring the methylene blue. So you have to remove the excess stain. It means excess color should be removed with the help of blotting paper or tissue paper. Okay, there should be no air bubbles. Means after placing the cover slip, air bubbles should not be present between the cover slip and also slide. Okay, that is another care should be taken. So observe temporary mount under the low and high power microscope. Low power means there is some objective lenses and ocular lenses. So these lenses or glasses should be low power. First you have to observe under the low power. After the observation of the low power, then go for the high power for clear vision or clear observation. So after, along with the observation, so draw the diagram in your notebook. Okay, this is another task. Okay, so this is about observation. Based upon your observation experiment, we have to answer some questions. The first question is, what is the shape of the cell that you have observed? The shape of the cell is roughly spherical. Okay, the shape of the cells is roughly spherical. And are the cell structures similar to the structure in onion peel cells? No, exactly not. So onion peel cells are going to look in this way. Okay, onion peel cells. Okay, but how the cheek cells are going to look like? The cheek cells are going to look like in this way. Okay, so are they going to look same? No, onion cells and cheek cells do not look same. It means the cells have different shapes and structures. Okay, is there any dark colored spherical or oval dot like structures near to the at the center of the cell? Yes, at the center of the cell there is a dark colored structure. This dark colored structure is known as nucleus, almost central, and there is a oval dot, dark colored oval dot is there. That oval dot is nothing but a nucleus. Okay, so to observe the nucleus, we need electron microscope. We cannot observe clearly under normal microscope or compound microscope. If you want to observe the nucleus clearly, we need electron microscope. Okay, so the nucleus is the largest main cell organelle. Okay, in the entire cell, the nucleus is only the cell organelle that is very large, and it is a main cell organelle. Why it is main cell organelle? Due to the presence of the DNA. Let us see the DNA, how it is going to look like. So this is a spherical structure of the DNA. This is a spherical structure of the DNA. It is going to have the genetic information. Okay, it is going to have the genetic information. This DNA is mainly responsible for the human behavior, human character, human height, human weight, intelligence, everything. This DNA is most important. So where this DNA is present? This DNA is present in the nucleus. So where this nucleus is present? The nucleus is present inside the cell. Okay, so in this way. So this DNA is most important. This DNA is a genetic material. DNA means deoxyribose nucleic acid. It is present inside the nucleus. So it is also control room. Why we are calling it as a control room? Why? Because this DNA is going to control what the cell has to do at particular time. Okay, when the cell has to divide, when the cell has to produce the proteins, when the cell has to produce hormones, when the cell have to respond, everything. So everything information is given by this DNA only. So this DNA is present in the nucleus. So due to this reason, this nucleus is known as control room of a cell. So the nucleus appears as a dark colored dots when stained with methylene blue. So during the process of the experiment, we have used a dye. Dye means what? A color. So that color is nothing but methylene blue. When you add methylene blue to the cells will take up the methylene blue and they appear as a dark colored structure. It means dark blue in color. Okay. So one important scientist, he is known as, his name is Robert Brown. Robert Brown is a scientist. In the 1831, he named the cell organelle as a nucleus. Okay, he named this as nucleus. Okay, his name is Robert Brown. So another scientist is Sleeden. He proposed the cell theory. So discovery of the nucleus was done by the Robert Brown, and cell theory was given by the Sleeden, and Sleeden called the nucleus as cytoblast. Okay, why he called the nucleus as cytoblast? In those days, the people thought that the new cells were created from the nucleus, but is it true? It is not true. But we came to know that the cell division is not only occurring from the nucleus but also cell. The cell division will be done by the cell along with the nucleus. So as a result, he called the nucleus as a cytoblast due to the thinking that due to thinking that the cells were created from the nucleus, but it is not 100% true. Okay, most of the eukaryotic cells have the nucleus. What is meant by eukaryotic? There are two types of cells. So two types of cells. Okay, what are the two types of cells? One is prokaryote. Okay, one is prokaryote, another is the eukaryote. Okay, so prokaryote and eukaryote. Prokaryote means they do not have the nucleus, no nucleus. Okay, so that is known as prokaryote. Eukaryote means yes, nucleus is present. Okay, nucleus present. Okay, so due to the presence and absence, the cells has been divided into prokaryotes and eukaryotes. We human beings and also plants, animals, all comes under the eukaryotes. Why? Because we have a nucleus inside the cell. But if you observe some bacteria, the bacteria comes under the prokaryotes. Why? Because the bacteria cells do not have the bacterial cells do not have the nucleus. Okay, due to this reason, we are differentiating the cells mainly on the nucleus. Okay, prokaryotes and eukaryotes. There are other categories also, means based upon some other factors also we are differentiating, but based upon the nucleus we can divide the cells into prokaryotes and eukaryotes. So here the most of the eukaryotes have the nucleus. Almost all eukaryotic cells have the nucleus, but few eukaryotic cells do not have the nucleus. What are they? RBC in mammalian cells. In our body, there are RBC cells. Our RBC cells do not have the nucleus. So RBC cells, no nucleus. Okay, no nucleus. And also plant sieve tubes. In the plant, there is a phloem sieve tubes are there. Not receive the sieve tubes. SCIVE sieve tubes. The phloem sieve tube cells and RBC in mammalian cells do not have the nucleus, but during the early development of the cell, during the early development of the cells, they have the nucleus. But when these cells mature, at that point of time, the nucleus will get damaged or nucleus will get disappeared. So hence we can say them as these cells have nucleus in the beginning of their life, but they will get destroyed in the later. When the cell is newly created, it will have the nucleus, but when the cell becomes mature, the nucleus will get destroyed. Okay, so the functions are, it regulates and controls the cell functions. Okay, all cell functions will be controlled and regulated by the nucleus. It determines the characteristics of an organism. What is characteristics means how many hands should be there, how many legs, how much tall, what is the color of the skin, what is the color of the hair, what is the color of the eyes. These all are the characteristics. You have the characteristics, your gender, your skin color, your legs, okay, length of your leg, length of your hand, your height, weight, everything. So these all are the characteristics of your body. So these characteristics are governed by who? These characteristics are governed by the nucleus only. Okay, So it consists of genetic information. Nucleus contains genetic information that involves in the processing of the cell division. All eukaryotic nucleus contains a membrane. What is that membrane? Nuclear membrane. Okay. So nuclear membrane is present around the nucleus in all eukaryotic cells. But in prokaryotic cells, there is no nucleus. So no nuclear membrane. So as a result, the DNA is freely floating inside the cell membrane, inside the cytoplasm. Okay. So along with that, just now we have discussed there is small holes on the nucleus. They are known as nuclear pores. Okay, they are known as nuclear pores. Example: animal cells and plant cells are the eukaryotic cells. They have the nucleus. So here the prokaryotes do not have the nucleus, and also the genetic material is free in the cytoplasm. Example for the prokaryotes is cyanobacteria and also bacteria, etc. So these all are the important functions of the nucleus. And I hope this lecture is helpful to you, and if you have any doubts or questions, feel free to post in the comment section and we will respond to your question and doubt. Okay, and we will discuss the next topic in the next lecture. Okay.